Nerve fibers. Definition of nerve fibers. A nerve fiber is a thread-like extension of a nerve cell known as axons. They are very thin transmission lines that run from each nerve cell in the body to receptors in the skin, muscles, and internal organs. They are the main information lines of the central nervous system, though these fibers are microscopic in diameter and can be very long. Properties of nerve. 1. Conductivity. The transmission of an impulse along a nerve fiber. 2. Excitability. The ability of a nerve cell to respond to a stimulus. 3. Ability to generate electrical events, nerve impulses. Nerve conduction. Nerve conduction velocity is the speed at which an electrochemical signal propagates down a neural pathway. Many things can affect this, including axon diameter, myelination, the internal resistance of the axon, and temperature. These impulses are extremely fast, with some myelinated neurons conducting at speeds up to 120 meters per second. Laws of excitation, conduction in nerves. Bilateral conduction. Axon conducts excitation is spread in both directions, one direction from dendrites to transmitters. Anatomical and physiological continuity of fibers. Intact, without any pathological problems. Isolated conduction. In peripheral nerve, impulses are conducted along with fibers independently. This is important because nerve trunk combines a large number of different nerve fibers which innervate different organs and tissues. Resistance of intracellular fluid is much lower than resistance of nerve fiber membrane. The nervous system is involved in everything the body does, from regulating your breathing to controlling your muscles and sensing heat and cold. There are three types of nerves, or neurons, in the body. Autonomic nerves. These nerves control the involuntary or partially voluntary activities of your body, including heart rate, blood pressure, digestion, and temperature regulation. Motor nerves. These nerves control your movements and actions by passing information from your brain and spinal cord to your muscles. Sensory nerves. These nerves relay information from your skin and muscles back to your spinal cord and brain. The information is then processed to let you feel pain and other sensations. Main difference of myelinated and unmyelinated nerve fibers. According localization, myelinated fibers are responsible for innervation of skeletal muscles, muscle spindles, tendon, and skin for fast pain response, as well as sensory organs. Unmyelinated fibers are responsible for innervation of internal organs and response to slow pain in skin. According to the velocity of propagation, myelinated nerve fibers are very fast, and unmyelinated nerve fibers propagate in a slow rate. The diameter of myelinated fibers is large, and of unmyelinated nerve fibers is small. The myelinated nerve fibers also present myelin sheath, but in unmyelinated nerve fibers, myelin sheath is absent. Let's watch a video about myelinated and unmyelinated nerve fibers and the propagation of impulse in each type.
What you will see and hear in this presentation is the development of myelin in the peripheral nervous system and the propagation of the action potential along a myelinated axon. This multimedia presentation will be most helpful if you already have a good understanding of the Schwann cell and the electrochemical process of the neuron, called the action potential. The Schwann cell forms a protective covering around the axon. Schwann cells start to develop in the embryo and continue to increase the wrapping around the axon through childhood. This development increases the thickness of the wrappings, which peaks in adolescence. This is why teenagers have such quick responses. The Schwann cell contains the typical cell organelles and cell membrane structure. However, notice as the Schwann cell surrounds the axon that the nucleus and other organelles are squeezed to the outside wrapping of the cell. This outer wrapping of the Schwann cell is called the neurolemma. The inner lining is made up of layers upon layers of cell membrane. This inner wrapping is called the myelin sheath. You will recall that the cell membrane, called the fluid mosaic model, is made up of a bilayer of lipids integrated with proteins. The thicker the myelin, in other words, the more layers of cell membrane making up the myelin, the more advantageous it is to the axon. One advantage is the regeneration of severed axons. Another advantage is an increase in the speed of the propagation of the action potential along the axon. The rest of this presentation will concentrate on the increased speed of action potentials down the length of the myelinated axon. Here is the neuron, and you can see the repeated Schwann cell membrane forming the myelin. Note that there is a small space between the Schwann cells where the axon is not covered by the neuroglial cell. These spaces are called nodes of Ranvier. From what you already know, action potentials occur at the axon hillock and continue to be repeated away from the cell body, much like dominoes falling one after another. An action potential starts on a polarized membrane which is -70. A stimulus causes the sodium gates to open slightly and sodium starts to trickle into the cell. If the cell reaches -60 or threshold, the sodium gates open wide and sodium floods in, bringing the inside of the axon to +30. At this point, the sodium gates close and potassium gates open. Potassium starts to pour out of the cell. This allows the neuron to become polarized again. Then the sodium-potassium pump starts to actively transport sodium out and potassium back into the neuron. First, we will look at the propagation of the action potential in the unmyelinated axon. Propagation is the repeating of action potentials down the axon. The action potential is repeated because as the sodium comes in, it diffuses to adjacent areas within the axon. As the sodium increases in this area, threshold is reached. Sodium gates open wide, sodium rushes in, causing depolarization and an action potential. As the sodium enters this area, it diffuses through the axoplasm and another action potential is created. This continues down the length of the axon. Now look at the myelinated axon. The same process applies to the myelinated axon. An action potential develops, and as the sodium comes in, it diffuses through the cytoplasm of the axon. It continues to diffuse through the portion of the axon wrapped in myelin. The increased sodium concentration reaches the node of Ranvier, increases the axoplasm to -60, and depolarization occurs. The sodium gates open wide, sodium floods in, and we have an action potential. Again, the sodiums diffuse through the axoplasm, reaching the next node. An action potential develops. The process is continued down the myelinated axon, passing from node to node. Compare the unmyelinated axon with the myelinated axon. You can see that action potential reached the end of the myelinated axon more rapidly than the unmyelinated axon. The speed of the propagation is faster going from node to node than action potentials that develop adjacent to the previous action potential.
General classification of nerve fibers. In the general classification, the fibers are divided into types A, B, and C, and the type A fibers are further subdivided into alpha, beta, gamma, and delta fibers. Type A fibers are the typical large and medium-sized myelinated fibers of spinal nerves. Type B are also myelinated fibers of the sympathetic nervous system, as well as preganglionic fibers. Type C fibers are the small unmyelinated nerve fibers that conduct impulses at low velocities. The C fibers constitute more than one-half of the sensory fibers in most peripheral nerves, as well as all the postganglionic autonomic fibers. The larger the diameter, the greater the conducting velocity. Alternative classification used by sensory physiologists. Certain recording techniques have made it possible to separate the type A alpha fibers into two subgroups. Yet, these same recording techniques cannot distinguish easily between A beta and A gamma fibers. Therefore, the following classification is frequently used by sensory physiologists. Group 1A of muscle spindles average about 17 microns in diameter. These are alpha type A fibers in the general classification. Group 1B fibers from the Golgi tendon organs average about 16 micrometers in diameter. These also are alpha type A fibers. Fibers from the annular spiral endings. Group 2. Fibers from the most discrete cutaneous tactile receptors and from the flower spray endings of the muscle spindles. Average about 8 micrometers in diameter. These are beta and gamma type A fibers in the general classification. Group 3. Fibers carrying temperature, crude touch, and pricking pain sensations. Average about 3 micrometers in diameter. They are delta type A fibers in the general classification. Group 4. Unmyelinated fibers carrying pain, itch, temperature, and crude touch sensations. 0.5 to 2 micrometers in diameter. They are type C fibers in the general classification. Methods of investigation. EMG. An electromyogram measures the electrical activity of muscles at rest and during contraction. Nerve conduction studies measure how well and how fast the nerves can send electrical signals. NCV. Nerve conduction velocity is a test to see how fast electrical signals move through a nerve. Now, let's take a look at EMG and NCV methods of investigation.